This Week in Parasitism is brought to you by the American Society for Microbiology at microbeworld.org/TWiP. This Week in Parasitism, episode number 46, recorded on October 16th, 2012. Hi, everybody. I'm Vincent Racaniello, and with me is Dickson Despommier.
Hello, Vincent Racaniello.
How are you doing, Dickson?
Doing very well, thank you.
Haven't seen you in a while.
Yes, you have. I was here yesterday.
Not on TWiP.
No, not on TWiP.
1 month.
Has it been a
Too
month?
long. It's your fault.
Right. I always get blamed for this.
You should. Today we have a special episode.
Yep.
A bit unusual, right?
It is.
We have two guests. Now, we're doing a paper today, and normally you and I would just do it.
We would.
But since the authors are right here...
No, and since we didn't understand it...
You speak for yourself. Dixon, you can speak for yourself.
I just did.
We're going to have the authors of this paper tell us all about it.
Yep.
So we should take advantage of that whenever we can, right?
Absolutely.
Dixon?
You bet.
So the name of the paper is Site-Specific Genome Editing in Plasmodium falciparum using engineered zinc finger nucleases. And the two authors are with us today. The first is Judith Straimer.
Hi.
Welcome.
Thank you.
Did I pronounce your name correctly?
Yes, you did.
Straimer. Very good. And the other author is Marcus Lee.
Hello.
Welcome.
Thank you.
Welcome to both of you.
Thanks for having us on.
You guys are both postdocs, right?
Yes.
Yes.
And they've come such a far way to get here, too.
We appreciate it.
Two flights of stairs.
Yes.
We're both from the Fiddock Laboratory, David Fiddock, right? And we have had David on TWiP. Do you remember, Dickson?
I do, of course.
When we did that. That was a lot of fun.
Yep.
So you guys are postdocs. Where did you get your PhD? How about you, Judith?
I'm from Germany and got my PhD at the University of Heidelberg in south of Frankfurt.
What did you work on?
On malaria too, on Plasmodium, so I stayed with it.
And then you came here as a postdoc to work with David?
Yes, last year in May I moved here.
So you haven't been here that long,
No,
right?
not that long.
And when you're finished, are you heading back? Or are you going to stay here?
Probably, but for now I'm really enjoying it. So
yeah. How do you like the U.S.? Good?
I love it. It's great. It's a very good experience.
Good. And Marcus, how about you?
So I did my graduate work in Australia, in Melbourne.
Did you?
Yep.
Wow.
Working on plants. I'm actually quite a recent convert in some ways to malaria.
Plasmodium falciparum has plant
almost
genes in
a plant,
it.
right?
Yes, it's almost a plant.
Just never see the light of day.
So is it falciparum or falciparum?
It's a bit like Missouri. I think
Doesn't
both
matter.
are probably.
Where in Melbourne? Notice I said Melbourne.
Yes. So I started at University of Melbourne in the botany department, and then we actually moved to La Trobe University, which is also on the outskirts.
David was in Australia for a while, right?
Yeah, he is Australian.
Is that the connection?
No.
Not at all.
No, and strangely, Melbourne is a really big hub for malaria research.
So it is.
Even though I wasn't working on it then, but I guess...
I did my sabbatical at Walter and Eliza.
Oh, yeah. That's
with David Kemp.
Fantastic place, yeah.
Yeah, I had a great...
Marcus, how did you end up in David's lab? You wanted to do malaria?
Yeah, so actually then I went to... This is my second postdoc.
Oh, okay.
So I went to Berkeley to do a postdoc in protein trafficking in yeast. The proteins we were working on are for transport from the ER to Golgi, and they're called COP2 proteins. They make vesicles which transport protein. And I came across some papers that described the potential role of COP2 proteins in trafficking malaria proteins outside of the red cell surface.
Okay.
And so that was really interesting. I think that's how I started.
Like the histidine-rich protein?
Yeah, some of those, and the EMP1, the VAR gene products that are involved in cytoadhesion. So curiously, in fact, it turns out that COP2 proteins are probably not involved, and the whole thing might have been some sort of artifact. But at
least it got you dragging
that's
and kicking
exactly right.
into malariology.
Yeah,
right?
and there are many, many interesting protein trafficking problems in malaria.
Sure.
So I have a connection with COP2.
You do?
How?
COP2 vesicles are thought to be involved in replication of polio RNA.
Ah, okay.
So the virus induces the formation of vesicles in the cell, and they're thought to be derived from COP2 containing vesicles, but there's a lot of debate. Some people think they're from the autophagic pathway, other people think COP2, but that's the connection that I have.
Mm-hmm.
So that brings you here, and you've been with David how long?
A long time now, about 6 years at least. I think.
Really?
Yep.
That's a postdoc.
Yes. I was senior postdoc in many senses of the word. Yeah.
A near assistant professor.
But you are thinking of leaving soon?
Well, jobs are not that easy to come by, as you may know. And my wife has a lab here at Columbia also, and so we're liking it here in New York, and at David's lab's a great place to be, and so
That's true.
yeah.
2 jobs are tough to get, but if this is one of the cities, you can do it.
Mm-hmm.
My wife was trained as a parasitologist,
Right. Yeah.
but that was with George Cross, but that was many years ago, and she's since left parasites. But this was one of the cities we wanted to be in because you could get 2 science jobs.
Mm-hmm. Yeah.
Anyway, you'll be okay. So let's talk about your paper. First, maybe you could tell us what was the problem you were trying to solve by developing this technique. Maybe Judith can tell us.
So in general, Plasmodium falciparum is very hard to genetically modify. It takes very long to engineer mutations or to introduce genes into the genome to study function of genes. And usually it takes 3 to 6 months. And then we came across the idea that introducing a double-strand break with zinc finger nucleases would accelerate and make the targeting of genes more specific. So this was the idea, to get a more efficient and faster system to modify the parasite.
So why is it more difficult to genetically engineer Plasmodium?
That is not that well studied, I guess. It's just, it has been very difficult to introduce DNA first, because the DNA has to pass so many membranes. There's the erythrocyte membrane, then there's a parasitoris vacuolar membrane, then there's a parasite membrane. So all DNA we want to introduce into the parasite needs to cross these membranes. And then, I don't know, somehow the DNA repair or the modification is just not happening as fast as in other systems.
Because in a mammalian cell, you can put a plasmid in pretty readily.
Yeah.
It gets taken up, it's integrated, but that doesn't happen.
It doesn't happen now.
No, it seems like a very random process. I mean, they'll maintain the plasmid for a long time. I mean, months and months.
Mm-hmm.
So as long as you provide selection, it's happy to keep the plasmid. But the actual integration event is really stochastic, it seems like. And
So that's why it takes so long. You're waiting for a very random, low-frequency event.
Right, which may or may not be deleterious to the parasite, depending on what you're trying to do.
Well, the other thing to suggest here is that you've got multiple stages to deal with too, right? I mean, you've got the merozoite inside the red cell, but then you have these sporozoites from the mosquito. If you could start with the sporozoite, it might be easier, but then you have to have a liver cell culture technology set up to get the... So it's a complicated parasite no matter which way you look at it.
Yeah, that is true for sure. And we actually in the lab, we just work with one stage, with the blood stage mainly, because this is the pathogenic stage of the parasite, so it's more, and it's the easiest to work with actually, compared to the liver stage, as you mentioned, is more difficult, and the sporozoites are very hard to produce.
And the merozoites can't be isolated in pure form so that you could just work with them first, or can they?
You can, and so there are ways to do this. You take synchronized cultures, and you can filter them, pass them through a filter, and get these individual merozoites. But they only remain viable outside of the cell for minutes at best, and so you would have to work pretty quickly. And it's definitely something to play around with, I think. But
So Dickson, I have your life cycle here. Because
actually, it's
a lot of people may be lost in
it's their life
all
cycle.
No, this is Dickson
I've
Despommier's
just been
life.
likened to a protozoan, for gosh sakes.
So the erythrocytic phase is what you usually use for introducing DNA, right?
Yes.
So that's this erythrocytic cycle right here. So here are the erythrocytes in the blood vessel. You drew this, so you should know this.
Well, I drew part
And
of
they
it.
contain these red things, which are the malaria guys, right?
Those red things, yeah.
These are ring stages.
They are.
And that is what they're talking about. But merozoites are what's released from the liver cell. Is that right? And you asked them if they could transfect those.
No, actually, the merozoites come out of the blood in the blood stage. So at the very end, when you make daughter cells, they're called merozoites, and they burst out, and they only last in the bloodstream for seconds, probably, before they find another red cell.
What about these sporozoites?
I asked about that already.
You asked about that, so you can't do anything with those.
Well, you might be able to, but then you'd have to have a liver culture set up to get the...
Exactly.
But those are what the mosquito injects, right?
Yeah, that's right.
And so that's precisely why it's actually quite hard to work with.
So those would die.
Exactly.
Those would die if you put them in just a medium after.
I mean,
They do survive for some time. There are many people are looking at gliding motility of these sporozoites, so they put them on surfaces and look how fast they glide, in which direction, which sense, just to get an idea how the sporozoites get from the skin to the liver, because they have to invade. They have to get to the bloodstream and then be transported to the liver.
Okay.
So they do survive for some time in media, but they don't replicate.
Okay.
And a lot of that work is with the rodent version of malaria, because obviously it's a whole different ballgame if you have infectious mosquitoes with Plasmodium falciparum that can infect humans. So usually when people working with mosquito stages, it's the rodent.
Stage. Otherwise, in the laboratory, you're going like this a lot.
Very worried,
That's slapping my hand and killing mosquitoes, in case the listeners want to know what I was doing.
Yeah, they'll probably hear it, Dickson. So you said there was a problem with getting foreign DNA efficiently integrated in a mammalian cell. In fact, you mentioned in your paper there are a lot of the components of non-homologous recombination are not present in malaria.
They don't seem to be.
No one has found them. We
and the whole
don't yet.
genome is sequenced, so we should be able
Yes.
to find them, right? So in mammalian cells, those are what help get DNA integrated efficiently. So if you take a HeLa cell and you put a plasmid in, very efficiently you can get integration, and we do that for years, and it's based on having DNA repair pathways. So you can't do that with malaria, and as a consequence, it takes months to get modification and add DNA, right? But you guys do that in your lab anyway, even though it takes months, right? Because I've heard talks from...
Yes.
Yes.
Yeah, I mean, you work with what you got, I suppose, and as long as you're patient, and then you assume it will happen at some point, then you can do it. But
it's tedious, right?
It's tedious. Yeah.
So then the idea, you said, Judith, came to use zinc fingers, nucleases, to do this. Did that come within your lab, or did you look at other people using these? Because they're used for other systems, right?
Yeah, they've been used in many systems, in Drosophila in plants, in Xenopus, and also in mammalian cells.
Mm-hmm.
And then we had a collaboration with Sangamo.
So in fact, the way even before that, the way it started, I think, was that a former student in the lab, Sophia Jale, was working on different ways of optimizing recombination in the parasite. And one of her thesis advisors is Lorraine Symington, here in this very department, who is one of the world experts in recombination. And I think she had suggested to Sophie at one of her meetings that we should think about zinc finger nucleases. And Sangamo is one of the experts in that.
Yes.
So that's kind of how...
Can you give a simplified explanation as to why that's a choice method to use?
Zinc finger nuclease?
Yeah, exactly.
Yeah, explain how a zinc finger nuclease works. Maybe you could start.
So zinc fingers are proteins which can bind very specifically to DNA sequences. And by that, we can define a very exact target where the zinc fingers bind. They are then fused to a nuclease, and these introduced on a double-strand break. So it's very specific, and it's very efficient in the sense that we can introduce a double-strand break in the genome, and that triggers then the previously mentioned DNA repair mechanisms.
So zinc finger nucleases are artificial enzymes. They don't actually exist because we made them. Humans made them. Right?
Yes.
They're a combination of the zinc finger DNA binding sequence fused to a nuclease.
Yes.
And the nuclease you used, I'm going to make you say the name, Marcus.
I'll say it very carefully. It's the FOC1 nuclease.
Sounds like a bleeper to me.
You're the boss.
FOC1, FOK1, which is a restriction enzyme, right? And it will cut. Does it have a recognition sequence, or it's a very...
No, it doesn't.
Yeah.
That is provided by the zinc finger proteins. They provide the specificity to the sequence.
So this is brilliant. You can try. And so in mammalian cells, I know people have used that. For example, we did a paper on TWiP where they try and use those to excise sequences, proviral sequences, to remove CD4 coding region from cells to make them resistant to HIV, for example. There's all sorts of applications. It's really brilliant. So the idea here is to use that to somehow introduce genes into malaria. So how'd you go about doing that? Who was so were you working on this before Judith came to the lab, or did you both start at the same time? How did that work? Because your former student had gotten the idea. Did she start doing the project?
Yeah, and now she's doing a postdoc in Heidelberg, as it happens. Okay. And so I'd started doing a little bit just to kind of get things up to flying speed, and then Judith came along, and then we were.
So when you joined the lab, David said you should work on this, or
yes, that's how it happened.
And you liked the idea.
I really liked the idea, and yes, it worked out very well.
What did you do first?
So actually, the first thing that we tried was to use some pre-existing zinc finger nucleases that Sangamo had already made to GFP, and so it's a brilliant, it's just a really simple system because you can see fluorescent parasites because they express GFP. And so those are pretty easy to make, and we just integrated GFP into the genome to provide a target that we knew were going to get cut by these zinc fingers that they had already made. And so then the idea was to simply to put in the zinc. We discussed a lot about how to get the zinc fingers into the parasite, but essentially the way we ended up doing was to put them on a plasmid, get the parasite to express the zinc fingers, and then they would cut GFP. And then the parasite had to do what any cell is trying to do, which is to try and fix the double-strand break. And so we provided it with a template that had some homology regions to GFP, but fused and framed with a selectable marker that would disrupt the reading frame.
Which was?
DHFR.
Right. Or dihydrofolate reductase,
yes,
for those that
exactly.
use the proper
the human
English.
Yeah.
Thanks.
That's okay.
Yeah, and so the idea was then to just select parasites that were resistant to dihydrofolate reductase because they interrupted GFP, so they should be resistant and they should not be fluorescent anymore. Right. And that was the assay.
So did that work?
And that worked. And so the thing that I think we were quite relieved about, at least the thing I thought was going to be the problem, was that the zinc fingers are going to be toxic to the parasite, and that hasn't seemed to be the case.
So you were thinking they would be having off-target effects.
Cut the genome.
So this first zinc finger you designed was designed to excise GFP, so you had some homology to GFP in the zinc finger?
Yes.
How many bases do you need for specificity?
So they had 6 individual zinc fingers on each side, which bind each 3 base pairs. So we had 18 base pairs on each side for each zinc finger. On each strand, exactly.
So so you can put together basically a module of so you go to the catalog and you've got zinc finger modules that recognize specific sequences and triplets.
Triplets, DNA triplets. Yes.
And all the triplets you can select from, like 3U's, UAC, etc., or TAC, or do you have to be more generic?
Well, I think that's the real art to the zinc finger design at the moment, and why they're not very easy to make, at least in-house. That it's really, there's some rules about what each zinc finger will bind in terms of the DNA target, but then trying to put those together in a way that makes sense and gives you a very specific binding.
So believe it or not, Vince, I actually did some work on Plasmodium falciparum while I was in Melbourne in 1986. And I learned, my big lesson there was I learned that it's a very AT-rich protein.
Yes.
So that's a big problem here, isn't it?
It is. I mean, it wasn't for the eGFP. We used the GFP because this is not a Plasmodium gene. So we knew that these zinc fingers would cut and they'd be very efficient. But to target endogenous Plasmodium genes, it is more challenging for sure. So
the GFP is easy because you just look at the parasites and they're no longer green. And the control is without some aspect of the zinc, maybe without the targeting sequence. Would that be the control? To see the spontaneous loss of GFP?
Actually, without the zinc finger. So we have the same donor sequence that the parasite could feel free to integrate. but without the zinc finger to actually make that break, it just doesn't do it. We were culturing for 2 months and nothing happened.
Right.
Whereas with the zinc fingers, essentially it's a catastrophic event for the cell, and it will try to find homology to fix it. And so within less than 2 weeks, we were having, getting parasites that were 100%.
So how did you feel at that moment when you discovered that actually worked? Did you have a party and did you break out the champagne?
I was very excited. I realized this was a big thing, for sure.
So let me get this straight. So you have GFP integrated somewhere. You probably picked a locus in Plasmodium that would not be harmful, right?
Yeah.
And these are green. And then you design a zinc finger to excise it. So did you excise the whole GFP or just a small piece in the middle?
No, we didn't excise actually anything. We just disrupted it. We introduced the double-strand break and used then the upstream and the downstream region of the double-strand break as homology, and then the selectable marker was integrated in there, so this would just disrupt GFP. That's why they lost fluorescence, but we didn't delete any sequence in this case. In this case, that's true. In the very first experiment.
So the selectable marker is on a separate plasmid or in the same plasmid as the
It was on the same plasmid.
So that has homology to where you wanted to integrate in GFP, is that correct?
Yes, we had,
Yeah, sort of nestled between two regions of homology in GFP.
So what I find interesting is that the plasmid that gives you the protein, the zinc finger, is also the DNA that goes into the genome to do the repair. It's very efficient, right? Okay, so that was a proof of principle, but as you said, that was easy because the GFP is not AT rich. So what was the next thing you had to do for this wonderful paper?
Well, actually, even before we left the GFP, leave the GFP idea, the other thing that we ended up doing was to try and replace GFP with RFP. And so this goes back to your question of whether we replaced it. And in fact, we did an experiment where we didn't have a selectable marker at all.
That's rhodamine fluorescent protein.
Red.
Red fluorescent.
Red.
Yes.
Okay, red.
Yeah.
Sorry.
You're too fancy, Dickson.
I don't know. I'm an old school guy here.
And so one of the issues was whether we could get this to work efficiently without any selection, because some of the times you don't want to actually disrupt a gene with a selectable marker, or even if you did, you're relying on the endogenous activity of that promoter to drive a selectable marker, and if it's not very strong, then that approach is not going to work. So we were thinking of ways of trying to get this to work without direct selection, just to see what the natural efficiency is. And so we just replaced GFP with RFP, without any selection at all. And partly, I think, because we use such big regions of homology, about 3 kilobases either side of the red fluorescent protein, it worked really efficiently. About 60 to 80% of the parasites that were transformed changed GFP to RFP. And so it was it's actually quite attractive to see. You have this mosaic of green and red fluorescent parasites under the microscope, and most of them were red, which was very satisfying.
How neat.
So,
how are you introducing the DNA into them? By electroporation?
Yes, yeah.
Okay.
Yeah,
so chemical ways don't work to get DNA into Plasmodium?
No, although curiously, you don't even have to have the parasite there. All you have to do is we do normally, the way we do it in the lab is we have infected red blood cells, but in fact you can just put DNA into uninfected red blood cells and let the parasite invade normally, and somehow it gets taken up.
That's
and
interesting.
It's
Yeah.
not clear.
Very interesting.
Yeah, it's not clear at all why or how.
Well, I think cells are very good at taking up DNA. It helps them evolve. That's how they acquire genes, horizontal gene transfer. I mean, so there are viruses that can do that, but then there are free DNAs that go around from cell to cell, and I think we have a natural pathway of uptake, and that's why that's my guess anyway. So you could replace now a green fluorescent protein with a red one. And what's next?
And then next, we try to target an endogenous gene, and we decided to go for PFCRT. That's the chloroquine resistant transporter in malaria. That's
Why not?
Why not?
It's going for the jugular.
So in this, we've been worked extensively on this protein in the lab. It's a transporter which transports chloroquine out of the food vacuole, out of its way of action, mode of action. And we wanted to target this one and asked Samgangmo to design zinc fingers for this gene. And in the first step, we only tried to introduce the single mutation, whereas previously with GFP we introduced a whole selection cassette or replaced RFP. In this case, we just wanted to edit a single nucleotide in the genome. And we did that by using a sensitive parasite, so a chloroquine sensitive parasite, and provided the zinc fingers targeting PFCRT, and at the same time, on the same plasmid again, a donor sequence of 1 kb spanning the zinc finger target site and carrying one mutation which would confer resistance to chloroquine.
Right.
And then we selected with chloroquine for parasites carrying this mutation. And this was very efficient also.
Wow.
So you
Nicely.
introduce the DNA. You wait a bit. I don't know how long you have to.
Usually we wait 24 hours.
Okay.
We after 24 hours after electroporation, we start the selection just to give them the parasite some time to recover.
And then you find growth in the presence of chloroquine.
Yes.
Is that basically it? Because otherwise they wouldn't grow if they were sensitive.
Exactly.
And so this is exactly replacing the sequence because you have to get the protein. It's got to be in frame, and it has that mutation, which is your readout. That's beautiful, right? So that was another exciting
Yes.
because now you're doing it in the genome, which is AT rich.
How you know it
Yes.
really works.
Yeah, so I mean, this is a real credit to Sangamo, and in particular
Exactly,
our collaborators Fyodor Urnov and Brian Zeidler, and they did the hard part in some ways of designing the zinc fingers
Zinc fingers,
that work,
yes.
and that's not trivial still. But that's what they do. And,
So when you do this, when you design these, do you have any way of checking them ahead of time, or you just do the experiment and see if it works?
They do actually.
Yeah.
They check them in vitro in the yeast system and check for activity.
Okay. You actually make sure that they will target what you want to target. So use a plasmid target, I guess, or something like that. And
yeah, I think they have a growth assay that gives a readout if it's cut. And just to give a sense of how tricky this is still, I mean, I think if you were to design 15 zinc finger pairs to a particular region, I think usually, only two or three or one might work. So it's still
it's a little bit of
difficult
magic,
problem.
right?
Yeah.
But I presume at some point there'll be a catalog of
Well, that would be fantastic. Yeah, off the shelf.
right for any triplet. There'll
New
be
England Biochemical, I'm sure that.
Yeah, and there are other ways of cutting DNA. There are other systems, I think, being developed as well. And as far as the process is concerned, as long as you can generate a double-strand break in the cell, no matter how you do it, as long as it's specific, then...
So I'm reviewing this grant now, and I don't know anything about anything, except I would like to know what the biological significance of this work is. I would like to know where you're going to take this for possible control of malaria. That's a big question.
So in terms of disease, and so one of the great promises of zinc finger technology is that you can use it for a lot of medical applications. It's not just a tool for the lab, but there are diseases that require correction of a single nucleotide change, and that's one of the beauties of the system, is that you can just make a single base pair change without selectable markers or anything, potentially. And for malaria, I don't think anyone's imagining it as a therapeutic in itself. I mean, you could never imagine delivering these with enough efficiency to try and kill malaria.
How about releasing malaria that's got this and seeing if it replaces the one that's already there and therefore might not be as lethal or might change its pathogenicity in some way? I don't know. I'm just thinking about it here.
Yeah, well, I think the way that we are using it is to look at drug resistance determinants. And so it's either from the field or from in vitro selections in the lab. There are parasites that exist that are resistant to various drugs, and the question is how. And so one of the hard parts has been testing candidates to find out, is this really a determinant? And so now, if you can come in, and hypothetically, the chloroquine resistance transporter, if you can just change what you think is the critical amino acid, and either switch it from resistance to sensitive or vice versa, then
that shows you that you were right after all. Then
in a defined genetic background, you can just make that one change you think is what's going on, and that's I think where we envisage using it in our lab at least.
Okay.
So the last experiment you told us you used another selection. Have you done it without any selection?
Yes, we have then repeated the same experiment only with selecting for the zinc fingers, just for the presence of the plasmid, because we needed to ensure that the parasites who hadn't taken up the plasmid were killed, and that we just enrich for expression of the zinc fingers. And then we could see an 15 to 18% of efficiency of edited parasites. It is not 100%, of course, but it's clonable, so it's good enough for us.
It's not bad.
It's not bad. So we could clone out parasites and then pick the edited ones and analyze
Now, if you don't select for the zinc finger plasmid, you'll never see it. Is that right? Or is it
well,
too
if we
long?
don't select for the plasmid, then we would just get the wild-type parasites, the ones which didn't even take up the plasmid, would just outgrow.
They would overgrow.
Yes.
So when one merozoite becomes transfected, so to speak, or changed by your technology, and then it replicates inside the red cell, you get multiple merozoites afterwards. Do they all have the gene? Or have you had occasions where a couple didn't have it?
Well, we don't know. I wouldn't know how to...
I think once they're modified in the genome, it seems stable. I mean, they initially start with the plasmid episomally, and so those can get lost quite easily.
Yeah, that is true.
But once they actually have the change, then...
And you have no idea of whether it's transferred forward into the sexual stages, and then into the mosquito, and then back into the host again. You have no lineage to tell you whether that's going to be the case or not. Would you like to know that?
Well, we haven't done the experiment with the zinc finger modified lines, although we plan to for some things. And but I would imagine it would be, because for other conventionally modified lines, those genetic modifications are stable.
But during mating in the gut of the mosquito, there's a chance for exchange, right?
I see what you mean.
True.
But only if you give them different... well, yes.
Yeah.
If you start with a clonal line, then I think you should be in good shape.
They could get,
Right.
Yeah.
Okay. Because I guess not everybody is familiar with the life cycle of Plasmodium, but...
Absolutely, yeah.
But so the ones that live in the red cells are haploid, right? And the ones that are secreted by the mosquitoes are haploid, but there's this very short period of time when they're diploid.
Exactly.
And so at that very point, I was just curious as to what you thought might happen here.
Yeah, well, they could recombine, of course, that's what the stage is for.
Sure.
But if we use two clonal lines, then potentially they both have the same, then there's no way that they could...
But if they were in different parts of the DNA?
Then, there's always recombination happening, so we can't control for it, but it's true.
I was going to also ask you whether or not there are multiple copies of certain genes that zinc fingers might identify, which wouldn't be completely covered by your zinc finger constructs. And so the other genes would escape that sort of a genetic disaster for the mosquito, or for the parasite, rather.
Yeah, that is true. Plasmodium has a lot of gene amplifications, and so if you wanted to modify a gene which is there in 3 copies, then this would be very difficult. That is true. I don't know if we haven't tried.
Well, I thought about the chloroquine-resistant genes being multiple copies, though. Wasn't that one of the arguments as to why some strains are more resistant than others, because they have more copies of this gene that keeps throwing out the chloroquine?
There's this MDR, multi-drug-resistant transporter, and this is in multiple copies, but the one we targeted is PFCRT, and this is just a single copy, so this was easy. But
we are actually interested in modifying MDR1 by zinc fingers too, so we may get that. We may get to that point at some point.
Sure.
So you're always introducing DNA in a haploid form, right? So that allows you to have in 2 copies of your modified introduced gene eventually. Whereas in mammalian cells, it's very difficult to achieve that, because we put DNA in. Typically, we don't replace alleles, although you can. But even if you did, it would still be only 1 copy in the end. So this is advantageous that you can do that in the haploid, right?
We're lucky.
It's good.
No, luck favors the prepared mind. Remember that.
So from start to finish, how long did this paper, which just got published this month, right?
Yes.
All the work that went into that, how long did that take?
Well, exactly from May last year till now.
And we started playing around with it in maybe January, so before you came. But then we got serious when you came. So about a year and a half.
That's pretty
maybe.
short. That's
and the zinc fingers, they only got delivered in May, June, I think.
Wow.
We started.
That's pretty quick.
Yeah, if only everything works so easy.
Yes, if only.
Yeah, don't get too used to this.
No.
So are other malaria labs going to be using this? I assume that since it's published they will, but do you know of any that have started?
I think we have a few people asking, were interested when it got published, and recently on a conference, many people asked me about the possibility of using them. So I think people are looking into it, for sure.
I mean, that's the test of a good method, right? How widely it's adopted. It seems to me that given the advantages this have, it should be. Are you or others trying to improve it? Because there's always a better way, right?
Yeah, I mean, we're still playing around with exactly, I mean, these are more sort of finesse questions, but how much homology do you need to get your reasonable efficiency? What other modifications can you put in? How big an insert can you stick in there? Can you use oligos even, rather than plasmids as donor sequences? And so mammalian cells has been demonstrated you can actually just use an oligo, a short oligo, to make deletions. And so if you could do that, that would be easy because it would save you a little bit of cloning time. You could just order up an oligo.
Right.
It's some of those tweaks that we're playing around with at the moment. And then ultimately, it would also be nice to find, to try and get more zinc fingers to see if we can modify different loci.
Sure.
Is it possible to get your constructs into uninfected red cells?
Yes, it is.
Yeah.
It is.
Okay,
It's the same.
And then if you infect them, will the parasite take it up?
Yes. They said that before.
Yes.
No, you said they mixed them together and then it took it as it was going into the red cell. But I wondered whether the red cell had it already and then you go in and then there it is.
Yeah,
that's
works.
what it. Yeah.
That's what you. Okay, fine.
that's-
So
yeah,
that cleared that mystery up for me. Okay, so.
Yeah, we'd actually, in the very early days, thought about whether because another way of doing it is to actually just microinject RNA or even try and get protein in. And the fact that the zinc fingers aren't toxic kind of relieves some of that worry that we'd have to get rid of them very quickly. So that hasn't been an issue. But some systems do microinjection rather
If you
than...
were working with Plasmodium lofuri, you would have a problem. Why?
Why?
Because it's a parasite of duct cells, and duct erythrocytes are nucleated. They have a nucleus also. So you would now have a problem of your construct going into the duct nucleus rather than into the parasite nucleus. The advantage of Plasmodium lofuri is that it grows synchronously in the ducts.
It does?
Yeah. See, I was at Rockefeller for 3 years with Bill Traeger's group. I wasn't working on malaria, but I paid attention.
Okay.
And if he didn't come in on the weekend to do his experiment after having infected the ducks on day 1, this was now day 7 or 8, he comes in on day 9 and they're all dead. He comes in on day 8 and he collects as many parasites as he can possibly collect. So he lost a lot of weekends at home, I can tell you that. But the nuclei of red cells in birds is still in the red cell after they have matured.
Okay.
So thank God you weren't working with Plasmodium lofuri.
Well, it may not have been a problem because maybe it would partition. Maybe some would go in the red cell nucleus and some would be taken up in the parasite. Who knows?
Perhaps.
You would have to do it. But maybe people who work on, do people still work on this nowadays?
Oh, sure. Yeah, because of the
They'll try it.
synchronicity of the infection.
So because you have the zinc fingers to target this transporter, you're focusing on that in part, right? Are you both working on this, or have you diverged now that the method is done?
Well, we sit next to each other, so we do actually talk from time to time.
We do.
But we're doing slightly different things now. So I'm looking at another potential drug candidate.
Okay.
And looking to see whether the mutations that are generated in vitro by selections actually confer resistance.
Okay.
And again, it's a very big gene. It's a little bit hard to work with conventionally. So if you wanted to put on a plasmid, you couldn't. Again, the AT richness makes E. coli scramble plasmids once they get quite big. So zinc fingers were perfect for this too.
Yeah, I've moved on to some other zinc fingers in the lab, trying to generate, make different modification. One is introducing an ATB site in the locus just to generate a reporter line for gametocytes, and this is one project. And then I'm also looking into some other nucleases. You never know.
To be better than the
maybe
FOC
very
one. Yeah.
easier to generate in the lab actually, because the zinc fingers.
So you said you're introducing an ATB site. Can you explain why you'd be doing that? Because a lot of people don't know what that is.
It is a site which allows you to integrate easily a plasmid with a single site crossover, and we're using that to introduce in this case a reporter line where we can differentiate between asexual stages and gametocytes. Because in the early stages, when parasites commit to gametocytogenesis, they're not easily distinguishable from the asexual stages, at least morphologically.
Yeah.
So, and to be sure which, what they committed to, we're going to introduce a reporter with a red fluorescent protein and a green, and then we can easily see that.
I think that's how you originally put the GFP reporter in by an at.
Yeah,
exactly.
Right?
It's essentially the system we had before the zinc finger
exactly.
nucleases, and that we can use integrase to catalyze integration. But the thing with it is that it's only at a specific site, so you have to have a parasite that's modified with this landing site, the at B landing site, for about 40 base pairs.
Is this derived from lambda?
Mycobacteriophage.
Mycobacteria, okay. So it's a similar system, an integration enzyme and a target. And that makes it a little quicker than it would normally take?
Yeah, again, much quicker. It happens very fast, and you don't wait around for months, as we were discussing. And the only constraint is that it's at a defined site, so you can't target any gene. It's
and of course, to get the at B site in the first place, that takes 4 to 6 months.
So that was the slow part.
Right. Yeah, exactly.
So if you know you want to work on a certain area, you put the at B in, you're patient, and then you could do it quicker after that. Yeah.
And we're just putting it in a different gene now compared to previously.
So in theory, you could disrupt every gene and ask which are required and which are not, right? Because I'm sure that information isn't known at the moment, right?
It's not now, yeah.
No, I mean, that would be fantastic to have a resource like the yeast deletion collection where every gene...
How many genes are there?
About the same as yeast, actually. About 5,500, more or less.
That's all?
That's all. A lot of them are a little bit... they're
all unusual.
Somewhat unusual, yeah. And it would be fantastic. But the only constraint is the cost of making zinc fingers at the moment. That's the.
How much is each one?
Well, so here's the rub. It's about when you buy it conventionally from Sigma, which is where you have to go. It's several $1,000s. I want to say about $10,000
to target one
to make
single
yes,
gene.
exactly.
Which would be, as you said, 18 and 18, right?
That's a lot of money.
Wow, that's expensive.
More than your average NIH grant.
Why is it so expensive? It's just an oligo basically fused to a nuclease.
Well, it goes back to this idea of how difficult it is to design, I think,
and
you have to make several versions to find one that works.
So if you go to Sigma and you want to buy them, do they have a chart of sequences and matched up, or do you put in your sequence and then they generate?
Yeah, usually we send them up to 500 or 1 kb of sequence of the gene we want to target, and then they look at the sequence and find suitable binding sites for the zinc fingers.
So they're doing some kind of analysis, and then they make it, and they give you a couple of possible
exactly. Usually they give us they find 10 binding sites, and then they test them in their activity assay, and then send us 2 or 3 pairs.
But in the old days, that was the way it was for siRNAs too. Companies had these programs, algorithms, and you'd pay them a lot, and they would tell you what sequences. Now everybody has the program, and you just do your own. So probably one day,
hopefully,
that would be really nice.
Yeah, because it shouldn't be so expensive, right?
It shouldn't be.
If it allows you to do so much, it shouldn't be that expensive. There shouldn't be a barrier just because the company wants to become very profitable or be more profitable.
Right. So I have some other questions that are related more to the general biology of the parasite. And I just asked you for a dream list now, okay? If you could just conjure up what would be the essential ingredients for being able to follow most of the critical life stage processes from the single merozoite to the signet ring stage of the next level of merozoites. How many different proteins would you need to follow? Not all of them. To get a good picture of what's going on during digestion, production of the rope trees, the extra membranes, and because I was thinking that as you're talking, you're tagging certain genes, and then you're looking at the fluorescence of that's what you've got a complete rainbow spectrum of fluorescent probes. So if you had 10 different proteins in Plasmodium that you could simultaneously follow
I see what you're saying.
in culture over time, what would that movie look like? I can tell you, it would be fantastic. It would be like watching a rainbow, and you could get all the processes, and the parasites would be alive still. So you could make those constructs based on what you've just said.
Yeah, I mean, so I guess one of the things we've been constrained with for a while is the number of markers. So that's one of the limitations to how many different things you could tag. But yeah, in principle...
because they've done this for E. coli already. They've done real-time protein synthesis in E. coli. You can watch single molecules of proteins being made because they've labeled them with fluorescence of some sort. So here's this whole technology.
Yeah, well, so funny you should say that, because just this week, probably next week, someone in the lab, Philip Heinrich, is going to be setting up our microscope to do time-lapse imaging. So a heated stage and following the parasites. And we're just talking about the asexual stage, so the 48-hour from a merozoite invading a red cell to making daughter cells and bursting out. But even within that stage, there's different substages, the ring stage, trophozoite, different things are happening, different organelles are being generated.
You'll redefine the life cycle based on the expression of these proteins. You'll call it protein 1, and you'll have a name for that. You say, oh, we're at the cathepsin D stage of production of, because, I mean, that's how they digest their food, right?
Yeah, and so those come on at a particular time, for sure. And some of that's quite well known because the malaria transcriptome was worked out on an hour-by-hour schedule some years ago in Joe DeRisi's lab. And it follows this really beautiful pre-programmed cascade of genes coming on and off and on and off. And so the proteins have been less well characterized, and so that would be very interesting. But in terms of the gene complement, it's true. It does this beautiful cascade.
You'd use an atomic force microscope.
Okay, now we're getting. This is really fantasy.
No, it's not. No, it's not fantasy. That's the deal. It's already here. You just have to do it. I want to come down and help you.
Anytime.
Sure.
I want to look. Because, seeing these things happen, we have optic lobes that are about that big in the back of our heads. We all want to see these things. We don't want to just talk about them. We want to. Why else use a fluorescent probe unless you can see it happen, right? Yeah. Tony James is a good friend of mine, and I remember when he first infected Anopheles stevens eye, he targeted using a Drosophila promoter sequence, because that's the only way you could get it to work, to the eyes, the ommatidia of this organism, right? Wow. And he produced a fluorescent green protein.
Yikes.
Mosquito that looked like it could have swallowed Cleveland.
We have a lot of fans in Cleveland. Be careful.
Well, okay.
They're going to send emails.
Omaha.
We have a lot of fans in Omaha.
We do. And Warren Buffett lives in Omaha. I won't touch that one at all.
So are there other parasites that are refractory to adding DNA that could be taking advantage of this? Toxo, can you get DNA easily into tox?
No, Toxo is perfect. It's very easy to manipulate. Yes, they don't need that.
Right. They invite it in.
Yes, they just inject some DNA.
How about your guy, Trichinella?
Trichinella is hard to do. You have to electroporate it through the cuticle, just like you do for Caenorhabditis. So it's tough.
But once you get it in, it takes it up readily?
Fine. It's absolutely fine. Yeah.
So there are no others that come to mind that...
Most of the good protozoan infections have been worked on, but entamoeba, I think, has not been used too much like this. The causative agent for amoebic dysentery, or...
What about Giardia?
Yeah, that's another. It has two nuclei. And they're both transcriptionally active, so you'd have to get into both at the same time. Like stick them in the eyes.
Interesting, yeah.
But that was a great question, Vince, because what other important protozoan infections have not been worked on like this? And the answer is none, right? American trypanosomiasis, African trypanosomiasis, those genomes are known now. So
Leishmania, we can
Leishmanium,
do.
that's right. By the way, are there any viruses of Plasmodium?
No, not that we.
Not that we know.
Not that I know of. No.
Have people looked?
It was probably. I'm not sure. I don't know.
They have to
honestly
be.
Don't know.
Everything has a virus,
Oh, they've looked at this one extensively. And how many genes of Plasmodium falciparum are plant-like compared to the other 3 species that infect humans? Of that 5,000 genomes, have all 4 been sequenced?
Have all.
How many plant-like genes? That's
Now,
a
you
good
already
question.
have your PhD, so this is... So,
Well, so the
Vivax, Malaria, Ovale, and falciparum, of those 4 organisms, do we know all about their genomes?
Actually, I don't know what the
think
genome.
Vivax is
Yes, Vivax
correct
has
for
been,
sure,
yes, but the rest I don't.
Ovale and Malaria, I don't.
Or Knowles eye?
But they all have the apicoplast.
Oh, sure.
And so they all have those
Okay.
remnant plant-like
Right.
genes. And
Right.
that is something we haven't actually thought about targeting with the zinc fingers, because that is yet another problem. So within the parasite, you have to get through several membranes just to get to the parasite.
Sure.
And then the apicoplast itself has another 4 membranes around it. And so that genome is very small, and it mostly imports a lot of its proteins. So a lot of the genome has been transferred to the nucleus. But
Did you get the thing that just went over everybody's head?
The 4 memory.
Mitochondria has its own genome, chloroplasts has its own genome, and the apicomplex has its own genome.
Yeah, I think we talked about it with Boothroyd.
A little bit, but not putting them all together like that. So does this have a mitochondrion also?
yes.
And does it have a genome?
Yeah, yes.
Yeah.
Of course.
Yeah.
So it's animal and plant-like. Or there's no such thing as an animal and a plant, it's just life.
So the idea is that the apicoplast, the origin is a chloroplast, right?
Or a remnant of some
just
ancient...
like the mitochondria are ancient bacteria.
So
right?
it's a symbiont. It's an endosymbiont. It's
But it's got
integrated
both.
into the subcellular particle
Why does it need both?
family. Well, I'd ask them.
So what does the genome of the apicoplast encode? Do we know?
So it makes what are they called? There's a paper recently from Joe DeRisi's lab again that showed that there's really only one thing in the blood stage of the parasite. So there's a fatty acid synthesis pathway in there, but that's not actually essential, surprisingly,
Really?
for the blood stage.
It's a remnant.
Well, no, it's important for other stages.
Maybe
So the liver
mosquitoes.
stage,
The livers,
for
okay.
example, but not the blood stage.
I see.
And it's IPP.
I know.
Isoprenoids
Yeah.
are what it needs. And so you can actually supplement a particular compound, just one compound, and then ablate the apicoplast.
And it wouldn't matter.
In the blood stage, it's fine.
So maybe if you introduced the appropriate zinc finger that would target one of those genes, maybe it just gets in also at the same time as it gets into the rest of the parasite. Maybe it would work. You could just try it. Right?
Yeah, I mean, it would have to get through into the apicoplast. I think that's the
Exactly.
tricky part. But
That's fair.
I guess,
Wow. What a complicated life.
yeah.
And still so successful.
It makes ours seem so
Quite.
simple.
So what is in your future? So you now have this system where you can use it as your meal ticket. You could go anywhere and say, I'm going to work on this aspect of the biology. I presume in your next job you'll work on malaria, right,
I
Judith?
will, yes.
So you can take this and you can look at something totally different from what you've been doing and say, I want to do this. I have the tools to do it, right? So you've only been here a short time, so you're going to stay a bit longer, right?
I will, yes.
But what's your long-term goal?
Cure malaria? No, I don't know. No, I'll keep working here. I'll stay. I have a few projects. And looking at one interesting idea is also looking at, especially for CRT, looking at very different alleles, because we know that it's a very important gene for conferring resistance to many drugs.
Right.
So, and with this technology, we really can make many parasites with all the different alleles. So that is definitely something which we're looking into.
Right.
So,
Eventually, you'd like to go out on your own.
yeah, but I still
You
have
haven't
some
thought of it
time,
yet.
I think. No.
Okay. Maybe that's a better question for you, Marcus. Are you on the job market?
Are you hiring?
We, in this department, eventually we will hire another parasitologist to keep David company.
So actually, my first love, I think, is still protein trafficking, and I also have some projects on that too. And the parasite does some really unusual protein trafficking things, but even the basic stuff, the basic machinery that all eukaryotes have, is not necessarily well understood. And so one of the things I'm working on is a set of machinery that's normally involved in making multivesicular endosomes. And this is very, very conserved. In fact, it's even present in archaea, some remnants of it. And the interesting thing about Plasmodium is that it's lost most of the pathway that a human or a yeast would have.
Right.
And all that's kind of got left is what archaea have. And so it's kind of devolved back into the primordial functions of this machinery, and it binds to an organelle. It does something. If you perturb it, the parasite dies, but it's not clear at all what this organelle is doing. And so that's kind of what I'm playing around with at the moment.
The most interesting thing I've read about in the last week involved something of this sort, where how do you get collagen out of a cell? And the collagen molecule, the procollagen, is much too big to fit into the vesicles to take it outside. So it has to fold it up and bend it, and then take it outside. Then it unfolds almost like a pop-up tent.
Yeah, I mean, that was one of the interesting questions about the whole, with COP2 vesicles, for example, because they're a very defined shape and size, and the question is whether the coat always has to make an 80 nanometer vesicle, or whether it can be plastic in some ways and make elongated vesicles for collagen or other things, or algal scales or anything that's sort of big that needs to get out.
Exactly.
Yeah.
Well, you are now marketable, very marketable. Take advantage of it. This story just came out, and here in New York, there are many institutions. I mean, NYU has an old parasite, an old malaria presence right there.
They do.
The Nussenzweigs. They're still active?
Yes.
Finished?
Is Ruth still active? I don't.
You could
so.
propose
Victor,
to replace them.
Okay.
Yeah, but Victor.
Cornell, Rockefeller, Mount Sinai, and then the boroughs have places. So there's a lot of opportunities here.
There
and
are.
it's very comfortable to stay here. It's great here, but you have to move out. And this is a good time. Right, Dickson?
I would think.
Anything else from you, Dickson, before we wrap it up?
It's an ongoing, fascinating topic about malaria and how the work is done. And I think we've had several presentations on malaria at various levels. But this is, for my money, the most fascinating level. Because now you're envisioning life as the parasite. You are becoming the parasite. And that's how I conducted my own research like that from the aspect of the Racaniello worm that I worked on. I imagined myself as the worm, and then I tried to imagine what the problems are that I had to solve, and then I tried to go
Now I understand you.
You do. You actually do.
By the way, speaking of understanding, do you now understand the paper?
I do.
Okay.
I do.
It's really beautiful.
It's crystal.
Thank you.
Appreciate
Crystal.
having.
Well, we weren't the only two that worked on it. Of course, there
There's
were
more.
several people.
Yeah, let's.
Still. It was
A
great.
bunch of authors here. Let's read their names to give them all credit. We have Judith Straimer and Marcus Lee. Andrew Lee, not related, right?
No, it turns out there's quite a few Lees in the world.
Brian Zeitler, April Williams, Jocelyn Pearl, Lei Zhang, Edward Rebar. Rebar, you know what that is.
I do.
Philip Gregory, Emmanuel Linas, Fyodor Urnov, and David Fiddock.
So it's essentially us here at Columbia, Sangamo over in California, and Manuel's lab in Princeton.
Princeton, right?
Yep.
Lovely.
Yep.
Great.
Very
All
nice.
good, very good collaborators. I'll
Yeah.
say.
Do you guys attend Gordon conferences?
Yes, sometimes.
You do? This would be a marvelous takeoff at a Gordon conference. I once ran one on a variety of subjects, but we had Diane Worth's group from Harvard and Tom Wellham's group from the NIH sparring off as to what the actual resistance mechanism was for Plasmodium falciparum, and they were both right.
It's always the way with these.
And then they both
It's always
proposed
the way,
different
right.
mechanisms. They had wonderful proof. They were ready to bash each other's heads in, and then it turned out that they were both correct. Then what do? You shake hands and, go back to your labs and find out some more about that. So that's how it usually turns out, right? Nature solves problems in multiple ways.
There's one more connection. So I told you about the COP2.
Oh, I know.
Do you know what the second one is?
Ribosome.
Yes,
Ribosome.
I work on
peptide?
picornaviruses.
Yes,
That's
exactly.
So you use this 2A sequence from a picornavirus to express both red and green fluorescent protein from to make a fusion and then they would be separated. So you make a translational fusion, and then this 2A sequence generates 2 polypeptides. Okay?
That's a very nifty system because they're made in fairly stoichiometric amounts,
Right.
unlike some other systems like
So
ribosome entry sites.
This Dickson, this is a 4 amino acid sequence, NPGP, right?
You remember that?
Yeah,
That's
I do.
amazing.
I was thinking about it for many years. It's present in some of the picornas, not the enteroviruses, but the cardioviruses and the aphthos, I think. Ribosomes pause before the proline and then start again, and you get 2 proteins. So it's not actually processed, it's just a ribosome skipping.
Yes.
And no one really understands how that
I don't
works.
understand how it
We
works,
don't
but
know.
just glad it
It
does.
works.
Yeah, what is the signal for that to stop here? You know?
And if you just take that 4 amino acids and put it in any protein,
I'll be
you
darned.
get 2. So why did you pick this? What was it?
Well, so in fact, it'd been used by Sangamo to fuse 2 different zinc fingers. So you need 2 different zinc fingers to make a pair. The nuclease dimerizes, and so we knew we needed to get both expressed. And this would be the easiest way to do it. We just weren't sure whether it was going to work, whether the ribosomes in Plasmodium would actually recognize that and skip it. And so we played around with fluorescent proteins first, and turns out it does.
That's a good point.
It must be a very universal mechanism.
The falciparum ribosomes recognize this pause, as they say.
Yeah.
That's the broader implication. I wonder if it's been done in bacteria. I don't know.
Yeah, I don't know. I don't know.
So they
actually.
would recognize it in archaea, whether all ribosomes would. Isn't that amazing, this sequence? So, Dickson, normally in the picorna genome, you have the capsid proteins, then you have a 2A protein, and then the rest of the genome. In my virus is the polio, the 2A is a protease that cleaves the bond before it. But in these viruses, the cardios and aphthos, it's not a protease, the 2A, but it's a skipper. You get this skipping sequence.
Amazing.
And you only need 4 amino acids. So the reason I know this is many years ago, I was reviewing a grant, and they were talking about this, but they wouldn't show any of the data because they said it was proprietary, because they were trying to commercialize it. So we said, well, we can't evaluate it unless you show us. So they had to give us the data, but it was really interesting. So 2 connections there. All right. Shall we wrap this up, Dickson?
Yes, we should, Vincent.
This is This Week in not Microbiology.
No.
This is This Week in Parasitism, and you can find it at microbeworld.org/TWiP. We can also find us on iTunes, and if you like TWiP, please go on over to iTunes. You should subscribe there and leave a comment or a rating. It helps us to stay up on the front page of the Apple podcast directory, and more people will find us and hear about the wonderful world of parasites. We do like to get your questions and comments. You can send them to twip@twiv.tv. Judith Straimer and Marcus Lee are both in the Department of Microbiology and Immunology here at Columbia University Medical Center. Thanks so much for joining us.
Yeah, thanks for having us.
having us.
That's great.
Appreciate it. Your next big story, we'll have you back.
All right. Deal.
Dixon Despommier is at Racaniello.org, verticalfarm.com, and medicalecology.org. Is it org? Not
It's an
com?
org. No, it's not a com.
Thank you, Dixon.
You're welcome, Vincent.
Always a pleasure to have you.
Same here.
I'm Vincent Racaniello. You can find me at virology.ws. You've been listening to This Week in Parasitism. Thanks for joining us. We'll be back soon. Another TWiP is parasitic.